Welcome to Future Talk. On today's program, we're going to talk about the use of nanotechnology to develop new kinds of materials with a range of practical uses. I have two guests, Professor Yi Cui and Professor Sarah Heilshorn, both of them professors of material sciences and engineering at Stanford University. They're both members of the interdisciplinary Gabal Laboratory for Advanced Materials, and they both have their own laboratories staffed by graduate students, postdocs, and visiting scientists, where they do basic research into the properties of matter at the nanoscale level and try to apply that knowledge to real-world problems. Yi Cui's research is focused on energy storage. He's developing lithium batteries which can store much more energy in a much smaller space than the current lithium batteries, which could have a huge impact on everything from mobile electronics to electric automobiles. Sarah Heilshorn is focused on developing new biomaterials made entirely of engineered proteins for use in regenerative medicine to cure bodily ailments. And I'd like to welcome both of you to the program today.
Thank you.
Okay. Yi Cui, let me start with you. How's your progress in developing these batteries? I understand you're doing very well, and we might see something on the marketplace sometime soon.
Yeah, we have been making quite a bit of progress in the past 5-6 years. As you know, batteries now for high energy applications, cell phones and laptops and transportation is dominating by lithium-ion batteries. In order to improve lithium-ion batteries further, the key is the materials inside the batteries, which can store lithium ions, can store electrons. So we invented a new technology, for example, silicon nanowire technology that can store much more lithium ions, up to 10 times more than graphite carbon used in the existing technology.
I understand we actually have some slides which can demonstrate some of this. Can we see that first slide, please? Okay, yeah. So what are we looking at here?
So the slide you are seeing is the comparison of silicon nanowire, that's in the B, the figure B, compared to the traditional of using film, big particle, that's in figure A. For silicon, it can store 10 times lithium compared to carbon, but the volume expansion is too much. Those big particles are going to be broken, the film will be broken. And by using nanowires, their diameters are small enough. There is a space around this nanowire, they can expand without breaking, so you can charge and discharge these batteries for many, many cycles.
So to make this work, you have to solve a lot of very complicated problems.
That's right.
Okay, and we have another slide. Can we see the next slide, please?
So the next slide is now change the topic a little bit. Everybody knows that the battery safety is so important. And we see the plane can catch fire, you know, due to
batteries can explode, overheat.
That's right.
Okay.
And the cell phones, you know, the laptops, sometimes you see the news catching fires. So the main reason, you know, they can catch fires is the internal short during batteries usage. Now we have a new invention. In this figure you see right there in the bottom, we have this the middle dashed line, that's a nano layers of conducting copper sitting between the two battery electrodes with the positive sign and the negative sign.
So right they can't short out, the negative can't jump into the positive?
That's right, there's a separator right there, separate them apart. Now what happened right there is this red color dash coating. When there's this shorting happening, it touched the middle, and this middle layer will detect this shorting by measuring the voltage. It becomes 0. Now at this moment, the battery is still safe. It's not shorting all the way from negative to the positive, so to prevent battery catching fires.
But it doesn't fix the battery, it just warns you that it's no good.
That's right, doing damage. It warns you. Now the research ongoing in my lab is try to remove those shorting by doing some smart function in our materials.
Now I understand you have one more slide. This one is not related to batteries, but it is related to your nanowires, another application. Can we see that?
Yeah. What you are seeing right here is we try to accomplish a new type of water filtration technology which can kill bacteria and viruses with low cost, very fast, high efficiency. And given the problem now California have, we have the drought problem. We don't have enough water, so we need to recycle the water. This type of technology allow you to do is having nanowires which generate very strong electric field, and those filters, when you apply a small voltage, there's a strong electric field right there to kill the bacteria and viruses. And this work, when we started, it was really nice collaboration between my group and Sarah's group in the same building. Sarah bringing a lot of expertise from the biology side.
Mm-hmm. Now, in order to use this as a water filter, there are parts of the world where you have to clean millions and millions of gallons of water. Can this be produced cheaply at a large scale?
Yes, it can be. This filter we use very low cost copper mesh and grow copper oxide nanowires right there, very easy to do. And the filtration speed is very fast. That's our intention to get millions of gallons of water every day.
Now, I understand that you work with some pretty distinguished colleagues on your team. Stephen Chu, the Nobel Prize-winning physicist, who also served as U.S. Secretary of Energy, works closely with you. What kind of contribution does he make to your work?
It's indeed very nice to collaborate with him. Last year, he came back to Stanford University after being Secretary of Energy. And he and I sat down and talk and chat about what are the most important energy problems we need to solve, and what are the potential technology can address those problems. We come down to, energy storage is one of the most important one. Both of us throw down a few ideas we all think very important. We start to work together, co-supervise graduate students and postdocs. Indeed, early this year, we have some breakthrough technology coming out addressing some of the problems has been around for 50 years.
I understand that in nanotechnology, matter at the nanoscale level has a lot of very interesting and unusual properties that you don't see at a larger scale. Can you just give an example of one or two unusual properties that people might not be familiar with when you observe matter at a really small scale?
Certainly in the nanotechnology area, there are many examples. A simplest example I would like to give is you look at gold. It's a golden color. But once you go to the nanoscale, become nanoparticle, their color change. They can be dispersed in solution. You can see a red color showing up. That's very different from the big size scale.
Now, Sarah, you're also involved in nanotech research, but you have a slightly different angle. You're looking at biomedical products, things to use in the field of medicine. What kind of biomedical products are you working on?
Yeah, my group is really interested in trying to design new materials that can mimic the materials that naturally occur in our bodies. So throughout evolution, our bodies have evolved to have these really exquisite materials with these interesting properties down at the nanoscale. We want to try and understand how that works so we can recreate our own proteins that begin to mimic some of those functions.
What would be some of the practical applications? What sort of health issues would you address first?
Mm-hmm. So right now we have projects looking at peripheral arterial disease. So this is very common with diabetic patients. They have compromised blood supply to the lower limbs. So we're interested in creating new materials that would help them to regenerate new blood vessels in those tissues. And that's a collaboration with Professor Nan Huang at the Veterans Affairs Hospital in Palo Alto.
So you would inject cells or proteins into the body which the body would think was part of itself, and they would start functioning as if they were part of the body?
Yes, exactly. And the trick is to design the materials so that they can recreate some of those functions that healthy materials in our tissue would normally have. So cells are the smallest living units of our body, but in order to do their jobs properly, they have to be connected by a material that organizes them into a proper structure and gives them the right directions about what to do and how to function. So we want to make our materials that we can use to replace that tissue when it becomes diseased or damaged, and so it's no longer telling the cells properly what to do.
Okay. So you're creating new materials to put into the cell, and I understand you're also creating materials to help you get the new materials into the cell, because you can't just inject it, it'll all break down. How you carry it into there is very important, and I think you have some slides to demonstrate that.
Yes, I do.
So can we see the first of Sarah's slides, please? Okay, so what is this about?
Yeah, so in the top left panel, what I'm showing is a vial of liquid and a vial of gel. And that gel is actually one of our protein engineered gels that we've designed to help when you're doing cell transplantation. So cells, and in particular stem cells, are being looked at for a variety of different medical applications. But when you inject stem cells through a syringe needle into a patient, many of them die. And that's what the lower left panel is showing. We've stained these cells with either a red dye or a green dye. And when they die, they turn red. And when they're living, they turn green. If you just inject them inside of a liquid carrier, which is what most clinical trials use today, about half of them, almost half of them, die. In contrast, if you deliver those cells within a gel carrier, the gel acts like a cushion to help provide some mechanical protection to the cells as they go through the syringe needle and enter the patient.
And the gel is also an organic substance, isn't it?
Exactly. So this gel is made out of our engineered proteins. So it would be a transient, short-lived material inside the body. It would slowly degrade back into its amino acid building blocks and just be resorbed and recycled by the body naturally.
And I think we have a few more slides on this concept. Can we see? Okay, there's the next slide.
Yeah, so when we try to design these new materials from scratch, we begin by thinking what type of tissue do we ultimately want to regenerate, whether it be blood vessels, muscle, or nerve, for example, and what would be the properties that an ideal material would have? What type of mechanical or structural properties would you want it to have? How quickly do you want it to degrade after you deliver it to the body? And so we design different nanoscale building blocks that we then use kind of like Legos. We mix and match them together in new combinations to create a new functional material that we could then deliver to different tissues in the body to try and help spur on tissue regeneration.
Now, working in such a small scale, and you have special tools, because how could you even see what you're doing?
So you can see it at the nanoscale, and an individual protein you also can't see by eye. And so we use a lot of biochemical and biomolecular approaches in my laboratory that might be more common in a chemistry lab, actually, than a material science lab to try and understand what's happening.
Okay, and we have another slide which illustrates more about this. Can we see the next one?
Yeah, so to actually make these materials, proteins in our body are encoded within a piece of DNA, and so we use that same strategy to make our engineered proteins. We encode that information, the instructions for making a new protein in a piece of DNA, and we give that DNA to a bacterial host, and then that bacterium becomes our material making factory and makes a whole bunch of the material for us.
So you start with a little bit of protein that you take from a person, inject it into this bacteria which replicates it for you, and then you have a whole bunch.
And so what we are injecting into the bacteria is a piece of DNA. And it doesn't have to be DNA from a person. It's a completely new synthetic piece of DNA that we chemically synthesized ourselves. But the bacteria is able to make a whole lot of it. And it's a bacterial culture of fermentation, much as the way you would ferment beer, for example. It's a very similar process.
Now, is that to create the gel that carries the cells?
Exactly. That's to create that protein material that we then can fabricate into a variety of different structures. We can make them into hollow conduits if we're trying to make synthetic blood vessels. We can fabricate them into gels that would be injectable. We use them as coatings for bone screws. That was one of the images that was on the slide.
And I think you've got one more slide. Let's see this last one.
Yeah. So this is an example of how you can kind of put all the different pieces together. So if you, the little yellow star there is a neuron.
At the bottom of each circle?
At the bottom of each circle, exactly. There's a yellow star there. That's meant to be, to demonstrate a neuron. And neurons, as they grow and regenerate, they send out these long projections that secrete enzymes. And we want those enzymes to degrade our material so that the neuron is able to carve a little tunnel through the gel so that it can continue moving along the nerve to regenerate. And that bottom panel is an actual image of real living neurons. This was a laboratory experiment that we did. So this is a cluster of neurons growing within one of our protein engineered gels, and they're sprouting out these projections through the gel, and they're able to grow uninhibited and able to remodel this gel as they go along.
Okay. Now let me ask you this. How soon can we expect to see some products coming out of this?
Yeah, so for medical materials that would actually be placed in people, there is a long safety and regulatory process that's monitored by the FDA. But we are already beginning preclinical studies. Those are done in animal models of diseases and injuries. And that's the first step towards getting these things eventually into patients. So I would say if things go well in our animal studies, it's conceivable that in 5 years we could be to first in human trials.
And what's the hardest thing here? Is it engineering the proteins to your specifications, or figuring out what the specifications need to be to accomplish the objective?
I think both are really big challenges right now in the field. California is a real leader in regenerative medicine. We have the California Institute for Regenerative Medicine, and we're really beginning to learn a lot about these stem cells and how they can regenerate tissue, but we still don't really know what material cues or signals to give the cells to get them to unleash their full potential. So we're still trying to figure out what are the right properties to design into our material to coax the cells to do all of their regenerative function.
So you're doing a lot of basic physiology research, trying to find out how the body really works so you can figure out how to fix it.
It is very much an interdisciplinary set of experiments that we have to do. So we work very closely with colleagues at the medical school who are, for example, developmental biologists and disease physiologists, and they understand the biology and then they explain it to us, and then we try to incorporate that into our new material designs.
Now, I have some questions that could be answered by either one of you. For example, how do you decide what projects to work on? Do you have autonomy in your lab to work on whatever you want, or is someone telling you, this is what we want and this is what we're going to fund? How does that work? Either one of you or both of you could answer that.
Maybe I can go first. This is really good about being a professor. So we can choose the topic we would like to work on. So when I try to think about what to choose, oftentimes it comes down to what's the problem I want to solve. You know, I look into what's the most important problem the whole world is facing, whether through engineering, through technology, particularly through materials design. I can solve those problems. If I have a great idea, I'll pick those problems to work on.
And I would just add that for me, I also enjoy having a great team to work with. So when I also look at and try to identify which problems do I think are most important and that I can contribute to meaningfully, because my work does rely so heavily on collaborators at the medical school, I will often look around and see, okay, how can I assemble the best research team here on campus? And that also helps to guide my research direction.
Now, this field is so new, I would think that you would need a lot of very special tools and equipment to do this work. Do you have to invent all those tools yourself, or is somebody else doing that?
For my research, I tend to use a lot of large-scale facility. Those equipment cost $1 million, multi-million dollars. Stanford University has been really good in purchasing the tools to satisfy the faculty's needs. So I benefit tremendously from the investment in the university. And for some of the tools, we also build our own. So it's a combination of both.
Okay. What's the most difficult part of the research? What are some of the biggest problems you have to conquer or challenges you have to face in order to get the job done?
So I think part of it is breaking it down into manageable chunks and figuring out how can my skill set contribute to really solving this problem. So as an example, we have a new project that's a collaboration with Professor Giles Plant in neurosurgery at the Stanford Medical School. And he's very interested in spinal cord injury. That's a huge problem. And so what part can I do as a material scientist to help tackle that problem, and how can I design a question that is a true testable hypothesis so that I'm not just going out and trying lots of different variables. I have a clear understanding of the project and I'm trying to decide, can I design an experiment to test that hypothesis so even if my experiment fails, someone else can learn from my results and the whole field can benefit from that.
So is it kind of like what Thomas Edison said, 1% inspiration and 99% perspiration? You get a few good leads and then you keep eliminating the things that don't work until finally something does?
Yeah, that's pretty much true, yes.
We try a lot of things probably that don't work.
That's right.
Now you're both members of this Gabal Laboratory for Advanced Materials. What does membership in the Gabal Laboratory bring to you that enhances your work?
So this lab is a highly interdisciplinary lab. It indeed consists of faculties from multiple departments try to tackle the problem of focus on materials, new materials to solve big problems. So if you look at what type of faculties in there, there's people from material science, there are people from physics, applied physics, from also other engineering departments. It's called, at Stanford, it's called Independent Research Laboratory, and it's try to break the department boundary to bring different expertise together to solve big problems.
What's the most satisfying part of your work? I guess it would be when something works when it's supposed to.
It does. It's always fun to have an experiment work, but I would say it's equally fun when an experiment maybe doesn't quite work, but it yields some surprising data, because then that's when you really gain some new insight into how the laws of physics and chemistry are combining together to really make this material function. So it's more the surprising data that I wasn't expecting that for me is the most satisfying part.
Right, because you're not just doing engineering, you're doing basic research.
Yeah.
You're looking into the basic properties of matter at the nanoscale level, and what you discover maybe will determine what direction you're going to go into next.
Yeah, I also add a little bit, in addition to this very exciting research supplies, our profession, one important goal is to educate students and postdocs. So our product, not only the research, the technology, but also the people we develop, that's very exciting as well. That's a big source of satisfaction.
Now, speaking about graduate students and postdocs, I've heard that there aren't enough Americans going into science, and maybe we need to find ways to encourage more young people to take an interest. Is there anything you do in your lab that could give insight into how this can be made an attractive career choice for children still in elementary school?
So I don't go out to elementary schools, but I do occasionally go and visit high schools and give seminars, tell them about some of the current advances being made in material science and how bioengineering can be used to help cure diseases and injuries, and try and relate it back to what they're learning in their high school geometry and chemistry and physics classes. There are a lot of concepts that high school students are learning that we're applying every day in our research. And I think when you make those connections, that can help get them really excited about the idea of considering engineering or science as a major in college.
Well, I understand some of your work also involves stem cells, which are very important. These are cells that have not yet differentiated or become specialized, and maybe you can make them specialize in what you want them to specialize in. What kind of work are you doing in that area?
Yeah, so for a lot of regenerative medicine applications, stem cells are a potential source of new cells that we could use to replace damaged or diseased tissue. And so we work carefully with stem cell biologists to try and understand what those cells are capable of doing, how can we do it safely, how can we do it ethically. And of course, my own area is how can I design a material that will give the stem cell some instructions so that it knows what to differentiate into. That is, it knows what type of functions it should be doing.
And I think you've also done some other interesting work with batteries. I read that you created a battery made out of paper and ink, which is actually pretty successful at storing electricity. And when you crumple it up, it still works.
That's right.
How does that work?
So, you know, for a while, we really try to develop the battery that's flexible, you can bend, you can crash, and then make sure they don't break. But the battery also needs a liquid electrolyte. So paper turned out to be really interesting substrate material. You can put materials on, they stay right there, just like the ink. When you write on the paper, how the ink stay on the piece of paper. So we can put the battery materials on paper, they stay strongly, and put them together. We know paper can be folded, you can do all kinds of crazy things, they won't break. That's how the whole idea comes from. It turned out to be working really well.
Yeah. We just have about a minute left. Do you feel you have all the resources you need to continue this work indefinitely? Do you see a pretty bright path through this, or is there any risk that maybe the people who pay for this will run out of money at some point?
So I would say, well, it's been this society has been very supportive, but certainly now in the United States, the funding support is not as strong as before. So to do the science and engineering, so I would say if the investment can be make more, then this whole area will grow even better.
Okay, and on that note, we're going to have to wrap. I'd like to thank both of you for being here today, Professor Yi Cui, Professor Sarah Heilshorn, both from Stanford University. Thank you for watching. Visit our website, www.futuretalk.net. For Future Talk, I'm Marty Wasserman. We'll see you next time.